About a year ago, we launched NMesh, which is the Nottingham Materials and Environment Science and Heritage Laboratories, which is essentially a collaboration of people with capacities and capabilities across the University of Nottingham who can come together and work on heritage science. The Nottingham Museum and Gallery Service are one of our partners. They are very interested in some of the techniques that we have, and they have asked us to come and have a look at George for them.
George the gorilla was purchased a long time ago in 1878 at the Paris exhibition by the then Nottingham Corporation, who are now obviously Nottingham City Council, and who run this museum, the Natural History Museum. So we don't really know where it came from. We think it's West Africa, but if it was the Congo, it could be either a Western or an Eastern gorilla, because we have Western one side of the Congo River and Eastern the other, a separate species. I did wonder at one stage whether there might be some goat hair in there, because 200-year-old taxidermy, it's very hard to preserve skins, and I thought there must be some patches that didn't preserve very well and that were filled in with goat hair. So we'll have to wait and see what the genetics tell us.
We've had our ancient DNA team in to take some skin and hair samples from George. We've also had our proteomics team to come in, who are going to look at things like the proteins in his body, which can tell us a little bit about his sex, his biological sex. We also have some questions about whether George is one individual or maybe a composite individual. It's very common in taxidermy, if not all of the sample is perfect, to potentially use pieces from another animal. So it's really exciting for us to be involved in this, using all the different kind of techniques and capabilities to understand a bit more about him, because he's a bit of a legend in Nottingham.
We recovered 4 hairs from the taxidermied gorilla George at Wollaton Hall. So the first thing I did was prepare them for a light microscope. Light passes through them and you can have a look and see if there are any details for them. But it wasn't very conclusive. I transferred those slides to a polarizing microscope, had another look. They look beautiful, but again, no real additional information. So the next step was to prepare the hair for a scanning electron microscope, which is the next level up, using electrons rather than light to look at. We had to mount the hair on a small metal mushroom called a stub, place it in a sputter coater, which coats it with gold to a very, very fine degree, and then it's available to be visible under the microscope that's behind me. What we found was a series of patterns along the hair shaft, which would be caused as the keratin dries, as it emerges from the hair follicle. There's not a lot published about gorilla hair, but I was able to locate a different preparation, and they look like a pretty good match. We also have a number of hairs from different animals that are taxidermied and kept in the School of Life Sciences in the zoology collection, and it does look like it's, well, a hair from a primate from what we can see. We need the corroboration from the protein sequencing and the DNA analysis to give us a final answer, but from just the hair ultrastructure, it looks like we're dealing with a genuine gorilla.
What is protein biochemistry? Well, we as living organisms are made of lots of different molecules. We're made of DNA, we're made of RNA, metabolites and proteins are the most abundant molecules in the body. We've got something like 20,000 different proteins as humans, and protein biochemistry is essentially analysing those proteins and understanding how they work. So how do cells function? How does the life process work? So in the laboratory we analyse ancient proteins, and the first sample type, the dental enamel extract, what we've done there is used a procedure called solid phase extraction. And what that does is essentially it captures the proteins and the peptides that are in that acid etch. So we're purifying out all of the proteins and peptides from George's teeth, and the next step will be to take those peptides to the School of Chemistry, where we're going to use a technique called mass spectrometry. And what mass spectrometry will do in this case is look for specific peptides in the samples from George that are indicative of the presence of X and Y chromosomes. So unusually, a particular protein produced by the X and Y chromosomes has different sequences, and if we analyse that protein, we can determine whether the teeth that George has are from a male individual or from a female individual. For the second sample type, the skin, that's more exploratory. So what we're going to do there is try and solubilise any proteins that we can, just to get evidence that proteins or molecules have been preserved in George's samples. So using the technique of protein electrophoresis, we've confirmed that molecules are preserved in the sample of skin from George. So that's reassuring for the ancient DNA analysis, because that does depend on good sample preservation. In terms of what we can learn from the ancient proteins themselves, probably not a great deal just from the protein sequences, although theoretically it is possible to analyse these proteins called collagens, which are abundant skin proteins, and collagens do contain species-specific information. So it might be possible at some point in the future to analyse those sequences and also confirm that the skin sample we've taken is from a gorilla.
Archaeogenetics is an interdisciplinary approach where we use the tools of genetics to answer archaeological questions. The Ancient and Environmental DNA Laboratory is a dedicated clean room space which we use to isolate and analyse damaged and degraded DNA from a range of materials, including museum specimens and archaeological objects as well. So from George we collected a couple of hair samples and also a skin sample, and it's the skin sample that we'll be using primarily for the ancient DNA analysis. And we'll do that through several steps, but the first one will be using a proteinase enzyme to remove protein from the sample, various purification steps, and then that should leave us with a relatively pure DNA sample. From there we'll use a set of genetic markers which we'll use to amplify DNA from George's genome and then sequence that DNA, and then that should tell us something about his ancestry. So in the case of George, we'll be focusing on mitochondrial DNA, and there are two reasons that we're going to use mitochondrial DNA. The first is that it's high copy number, so it's an ideal in an ancient DNA situation where we're looking at damaged and degraded DNA. So just the fact that there are more copies available means it's more likely that we'll have success. The second reason is that there are parts of the mitochondrial genome that tend to be more variable than others, and it's this variability that we're going to use to work out which population or which subspecies of gorilla George belongs to. NMesh gives us real new opportunities to work together and synergise, to all donate, if you will, simple approaches that collectively give us so much power to get new insights into heritage science, into humanities research, but also into ancient proteins research as well.
So NMesh is interested in pretty much anybody who's got heritage science questions. So big organisations, local community groups, individuals. If you've got archaeological objects, if you've got historical objects that you're interested in finding a bit more about, then we have a team of experts who do all sorts of different things, so from ancient DNA and genetics, proteomics, archaeological investigation, all sorts of things to do with heritage science. If you have questions about any of those aspects, then get in contact and we'll see if we've got the right person at the University of Nottingham and through NMesh who can help you.